pancreas is the location where most digestive enzymes come from, and here we're going to talk about these pancreatic secretions. So some anatomy here. We have the pancreatic duct that comes through the pancreas and joins up to the common bile duct, which comes from this tree shape in the liver. The secretory functions of the pancreas are both endocrine and exocrine. Endocrine pancreatic secretions come from the islet of Langerhans, the alpha and beta cells in there, and the hormones insulin, glucagon and somatostatin are essential for the regulation of metabolism. Insulin is your main anabolic hormone, and glucagon then appears in the absence of it to regulate your blood glucose levels. The exocrine functions are both to secrete an aqueous component and an enzyme component. To separate the two different functions, both digestive and protective, the digestive functions of the secretions are the productions and secretions of digestive enzymes to further digest the food ready for absorption. But also there's a neutralization aspect to it. There's a lot of bicarbonate in these secretions that neutralize the acidic chyme and allow these pancreatic enzymes to function near their optimum pH. This neutralization process is also protective because the chyme that comes from the stomach is very acidic, and it needs to be neutralized rapidly in order to protect the duodenum that doesn't have these tight junctions and this protective mucosal layer that prevents the gastric acid from harming it. So as the contents of the stomach comes in, it's rapidly neutralized and then acted on by the secretions of the digestive enzymes from the pancreas. The aqueous component of the exocrine pancreas is secreted at a rate of about 1 liter a day, and as I said, it's rich in bicarb, and this increases the pH of the duodenum contents. And the rate of secretion of this aqueous component will alter its composition, and generally it's isotonic with the blood, but as secretion increases, you get an increase of bicarb secreted and less chloride. And so at maximum rates of secretion, it's essentially sodium bicarbonate, which is a very alkaline solution, very adept at neutralizing the acidic contents that's just been delivered into the duodenum. Similar to the cellular mechanisms of acid production, I want to show you the cellular mechanisms of bicarbonate production, because there's similarities to it, although it tends to be reversed in most cases. So it starts off with the same reaction. From metabolism, you get carbon dioxide and water created, and then carbonic anhydrase then stimulates the creation of carbonic acid, and then this dismutates into bicarb and a proton, and rather than the proton being pumped into the lumen, this time it's the bicarbonate portion that's pumped in using this bicarbonate chloride exchanger. The proton then is exchanged out through the basolateral membrane using the sodium hydrogen antiport, and also the potassium proton ATPase also pumps the protons into the blood supply. As the bicarb is passed into the lumen, this takes with it sodium and potassium that pass through the duct lumen, so it passively just enters in, and this whole flow of ions then creates an charge that then this chloride recycles through chloride channels into the lumen of the GI tract. In the stomach, we saw the alkaline tide as bicarb was pumped into the blood. Here we have sort of the opposite effect, which is the acid tide, because there's a net excretion of protons into the blood. And so if you were to measure the blood that comes from the pancreas, it's actually more acidic than normal because of this active pumping of protons into the blood supply as bicarbonate is passed into the lumen of the pancreatic ducts. There are several different types of digestive enzymes that are secreted into the pancreatic ducts. You have proteolytic enzymes to degrade proteins, amylolytic enzymes to degrade carbohydrates, lipases to degrade lipids, and then nucleolytic enzymes that digest nucleic acids. The main ones are shown here. Then we have both endopeptidases and exopeptidases that are secreted. You have alpha amylase, which is secreted, again, it was first secreted in the saliva, now it's secreted by the pancreas. A few lipases that separate triglycerides and cleave cholesterol esters. And then a lot of our food, our cells with DNA in them and RNA in them, and these nucleolytic enzymes break down these nucleotides into small molecules that then can be digested. Obviously, the problem with having proteolytic enzymes released is that they will cause bystander injury to the local environment if they're not activated at the right time. So proteolytic enzymes are generally secreted in an inactive form from the pancreas, the inactive zymogen form. And then you have enteropeptidases that are secreted by the duodenal mucosa, and these will activate trypsin from trypsinogen. And then trypsin will activate itself and the other proteolytic enzymes to go on and start digesting the proteins that are in the duodenum. There's a trypsin inhibitor protein in the pancreatic secretion that prevents premature activation of the proteolytic enzymes in the pancreatic ducts. The alpha amylase is secreted in active form because there are no carbohydrates that upon digestion would cause pathology. And also there's a colipase that is secreted in the pancreatic secretions, and this prevents inactivation of a pancreatic lipase, which would happen by caused by bile salts. And so this colipase protects the pancreatic lipase from this inactivation. The control of the pancreatic secretion is both neuronal and hormonal. Neuronal is by the parasympathetic input via the vagus nerve and cholinergic receptors, and this increases enzyme secretion. Sympathetic input causes vasoconstriction, and that inhibits secretion by lowering the blood supply. Hormonal secretion, secretin, produced by the duodenal mucosa, would stimulate bicarb-rich secretions, and then cholecystokinin from the duodenal mucosa as well will stimulate enzyme secretions, and then gastrin, substance P, and VIP will additionally cause secretion of pancreatic secretions, but these are sort of less potent than secretin and cholecystokinin.